Namaste. Myself Manjri Gupta and today I am explaining about the genetic markers. These are the traits or character that is polymorphic, easily and reliably identified and whose pattern of inheritance can be followed at morphological, biochemical and molecular levels. These can be classified into various types. The first is morphological markers. These are the visually distinguishing qualities. Next, the size, shape, and pattern and position of the chromosomes can be used as cytological markers. And third types are biochemical markers. And the molecular markers, they are specific fragments of DNA that can be identified within the whole genome and used to flag the position of a particular gene or the inheritance of particular characteristics. The properties of ideal genetic markers, it must be polymorphic, codominant inheritance, reproducible, etc. And the two types of markers, hybridization based and PCR based. This is based on the hybridization technique. It is expensive and require total cellular genome, whereas PCR is reduced time, efforts, and expenses, and a small amount of DNA is needed. The first is RFPL. This is a hybridization based techniques, and the workflow is the first, the DNA is isolated, then digest with the restriction enzyme and separated. The DNA fragments are separated using the agarose gel electrophoresis, and then they are denatured into single-stranded DNAs and then hybridized using the radio-labeled probe while transferring into the nitrocellulose membrane filter, and then the probe signal is detected. Next is polymerase chain reaction based markers. First is RAPD. It is a randomly amplified polymorphic DNA. In this, small primers are used to amplify the DNA sequences, and then they are visualized by using the technique agarose gel electrophoresis, and monomorphic or polymorphic DNA is identified according to their weight of the DNA. Next are simple sequence repeats. These are the small segments, usually 1 to 10 base pair long, and they are present at the various sites of the genes, DNA, and can be present at the coding or non-coding regions in the eukaryotes. And if they are present on the coding region, they can cause a genetic disorder. And those genetic disorders can be identified using the simple sequence repeats. And these are also used in the DNA fingerprinting and other techniques also. Next, the applications of molecular markers. These are the genotypic selection, indirect selection, quantitative traits of loci, marker-assisted selection, selection of recombinants from non-recombinants, etc. And these are the comparison of most broadly used techniques of molecular markers. This is, and next is, thank you.